It is also my pleasure to introduce our next speaker, Dr. Suzanne Bohlson, who is visiting us all the way from the Southern California chapter of SCS and is a professor at UC Irvine. She was our distinguished speaker at the Gold Mass yesterday as well. And we're grateful that she's been willing to give us a second talk here for our conference. Dr. Bohlson is on the faculty of UC Irvine, where she also serves as director of several graduate programs in biology and biotechnology. She specialized in immunology and innate immune responses, earning her master's from Washington University in immunology and a PhD in biology from the University of Notre Dame. However, before, she was Catholic. She has published extensively on leukocyte receptors and immune system function. She was raised in an irreligious academic family and converted to Catholicism in 2007 at the age of 36. A beautiful story she told us about last night after the Gold Mass. She has since written and spoken about the integration of scientific inquiry and Catholic faith. And she is a member and the president and founder of the Southern California SCS chapter, and today she will tell us about lessons from immunology for the journey of faith. So please welcome Susie Bohlson.
Thank you, Chris. And wow, how intimidating to speak after Martin Nowak. But anyway, I think the way he describes seeing God in mathematics is the way I see God in the immune system. And I hope to convey some of that, though it may look, again, like parallel thoughts instead of a cohesive thought, depending on your worldview. I'm going to try it. This is a new talk for me. Lucy gave me the option of not only speaking last night, but talking today. And so I'm trying something new and talking about more of my science, and we'll see how this goes. All right. So I think there are a lot of parallels between the immune response and the spiritual life. And that's my main interest is faith. As a convert to the Catholic Church who has taken to it in a way that is very different than the way I think about my science, but I'm as passionate about my faith as I've been about my science, trying to understand why, what makes me tick. And so a lot of that comes out in my talks. But anyway, I see the immune response like the spiritual life, and it's all about seeing the big picture and responding to it appropriately. And so, as in our spiritual life, where we try to recognize the environment, that's what our immune response is doing, and it's all about how we respond to environment. So when we think about the immune system, it's a brilliant system. So this is a system that has to be able to recognize all kinds of different pathogens. So pathogens come in sizes that we can't see under a regular light microscope, like the viruses. These kinds of pathogens that get inside of our cells and take over our own machinery. And pathogens come as bacteria that are seen outside of cells, bacteria that live inside of cells. Pathogens come as yeast that are bigger cells, eukaryotic cells that in some ways share a lot of things in common with our mammalian cells. Pathogens come as huge parasites that are much bigger than our cells. And somehow the immune system has to tell the difference between all of these different pathogens and respond appropriately. And the reaction it has to a virus is very different than the reaction it has to a multicellular worm, for example. And similarly, when we think about the spiritual life, a healthy spiritual life is going to protect us from sin or disordered love, however you want to think about that. I was in Japan when I was putting together this slide deck, and I Googled 7 deadly sins to try to get an image, and that's why we have the anime image of the 7 deadly sins. So in my work, I've been interested in understanding how cells in the immune system can recognize the environment and tell the difference between what you might classify as good and bad. So a cell like a macrophage, pictured here, has to be able to recognize whether it's looking at something like a bacteria over on the red side, or whether it's looking at something like a dead cell, which it also has to clear and get rid of effectively. So this is a cell type that eats stuff and gets rid of it in an appropriate way. So when we think about loss of dead cells, about 50 to 70 billion cells die by programmed cell death or apoptosis every day. This is a silent process, and the body just does this. It gets rid of old self or dead self. And this is done very silently. Somehow that macrophage knows that it has to get rid of it. It has to get rid of it fast, and does that very quietly. Versus a pathogen, it also knows it has to get rid of it and get rid of it fast, but sends off a lot of alarm signals to activate the body, and so you have to mount a different type of response. When the body doesn't clear the dead self or the apoptotic cells, it results in or contributes to what we ascribe to the waste disposal hypothesis. Those cells build up inside of us. They start to disintegrate. They start to release compounds. They start to look dangerous. They start to look like pathogens, and so we mount a different type of an immune response, and this is when things go wrong. And this is when we start to attack ourselves, and we end up with autoimmunity. And I think this parallels, again, to the spiritual life. I think maybe when people get to about my age, they really start to think about what they've done up to this point, how they've built themselves up, their career, all the things that required to build an ego. And then they look out at what they've done and realize that there's another mountain to climb, that we need to get rid of old self, and we need to start focusing on that self that is not ego. And so I see parallels between clearance of dead and dying self in the immune system, and also climbing that second mountain, getting and seeing the world without ego involved. And again, there's a lot of popular writers that are writing about this, like David Brooks, but also the mystics like St. John of the Cross, Teresa of Avila. So back to the recognition event in the immune system. So a lot of my work has focused on a group of receptors called pattern recognition receptors. It's how the immune system recognizes stuff that it's encountering. And these receptors are found all over the place. They're found on the outside of cells because some pathogens and things are on the outside. They're also on the inside of cells because things like viruses and intracellular bacteria are detected here. They're also on the cell membrane because a lot of things come in contact with cells, and that's how you encounter them. And so there are these receptors all over the place that allow us to recognize internally with our cells what we're seeing. And a molecule that I've spent a lot of time thinking about is a protein called C1q. And just within this one molecule that sees some things like dead and dying cells, there is so much complexity in it. So one molecule of C1q is a hexamer of trimers, so 3 peptides come together, wrap around themselves to form trimers, and then 6 of those come together to form a molecule that, if you can look at it by EM, looks like a bouquet of flowers. So there's a lot of beauty just at the molecular level of this single pattern recognition receptor that recognizes dead cells. And then you take that molecule, and we've done work to uncover multiple pathways by which that molecule alone can recognize dead and dying cells. So it's part of a system called the complement system that activates a proteolytic cascade that results in deposition of complement all over the apoptotic cell and rapid engulfment into the macrophage. C1q by itself can bind to a cell, regulate gene expression of that cell to turn on a pathway that shuts down inflammation but ramps up engulfment of dead cells. C1q also bridges that connection between the dead cell and the macrophage. So again, a molecule that has so much complexity in and of itself coming together then has multiple pathways by which it can dictate just this one single event of becoming a pattern recognition receptor that helps these cells to recognize what they're seeing and how to respond appropriately. And as scientists, I think to figure out these kinds of things and to understand how these pathways work, there's a type of direction that we have to take in our thinking as experimentalists in the laboratory. And I can illustrate that with an experiment that we ran where we were controlling, of course, the reagents, and all of that matters. You have to control what type of water you use. You have to know what's in your water. You have to know even to the level of what plastics you're using in the laboratory to start to figure out these kinds of pathways. And we knew that, and we had a phenomenon in the lab where we recognized, and we were studying, that some cells of different backgrounds rolled up on plates, and some adhered very strongly to plates. And that mattered because that property of adhesion is something that's important when you think about cells migration, metastases, engulfment of dead cells, all of those kinds of things. So it was a phenomenon that was worth studying. And one day in the lab, it stopped working. And so, we were getting close to publishing this paper, and all of a sudden we lost the phenomenon that we were trying to investigate. And it turned out that when we mapped it back, because we knew all the details of the reagents and supplies that went into the experiment, when we mapped it back, it turned out that the plastic that was being produced was coming from, initially distributed from a laboratory in Canada, and they switched the distributor, and now it was coming from the US, and that changed the phenotype of the cells. And so anyway, all of this to just point out that as scientists, and part of building up that self in the first part of our lives that allow us to be productive in the laboratory, part of it is just recognizing and controlling our systems so that we can do successful and reproducible science. Now bring that all back to trying to integrate both science and faith. So we're coming from places that have to regulate and control systems so that we can study them and investigate them and know everything at the molecular level and recognize that we can ask questions at just tremendous detail about all of these phenomena. And then we come into the church. And how do we use that thinking or work with that thinking in the church? In the church, it's often not a simple thing for a scientist to do. So an example out of that is one of the things I do at the church is I help with the retreat program. And this summer, we went on a retreat, and part of that was showing movie night, and we were showing Babette's Feast. I'm married to a guy that does movies for his hobby. That's what he loves to do. And so we have a little movie room set up in our living room. He would love this here that we're at a movie theater ourselves today. And me, with the detail orientation I have, I made sure that I brought 2 DVDs of the movie in 4 different languages with and without subtitles, copied onto my laptop, copied onto a USB drive, and the DVDs themselves. So if anybody could show a movie at a parish, it would be the 2 of us. And we got into the middle of that movie and got a blank screen. And no matter which format we tried to use, we couldn't play the movie. So as a control freak scientist, I could have locked everybody in the room and said, you got to watch this movie. We're going to figure it out. And we probably could have figured it out eventually. But what happened instead is we all went outside, because what else could we do? And that night at the mission where we were having the retreat, it happened to be them celebrating their feast day. And so we walked out, and as soon as we got outside, right about then is when the fireworks began. And if we just walked about 100 yards or so, we were able to walk into a festival where there were native dancers, priests that welcomed us into the church for adoration. And it was a festival like I didn't know it was coming. I had no idea, and I'm part of the retreat team that planned that. And so it was just one of those most favorite moments of the retreat for the parishioners, because it was something that was completely out of our control and something that everybody on that retreat saw as a gift from God. And so I think that God works in our lives in a way that we can't control, and that's part of what makes it so difficult, I think, as a scientist with faith, to bring these worlds together. And so this was my last slide from the talk yesterday. Again, that I think that living a life of faith as a scientist is similar, again, to the immune system. It's about recognizing your environment and responding appropriately. And I think that often we're trained in a way that makes it hard to do that as scientists. And for us, I think it takes a lot of courage to test those boundaries and what we can actually do with our faith in academic environments. And as I've told you, in these past few years, I've been able to bring these things together more than I've ever been able to do before. And sometimes it's just testing that and looking for green lights. But in the end, I think really our leukocytes are much better at it than we are at responding to the environment appropriately. And for us, there's just a lot of unknowns that we have to figure out, and with trial and error. So in conclusion, our immune systems are finely tuned to recognize the environment and respond to it appropriately. And I think it mirrors a healthy spiritual life in which we recognize the environment and respond appropriately. And I think that I've always been able to see God in my work because of those similar principles. And as scientists, we're required to understand complex details and control them in experimental systems. But as persons with faith, we have to give that up. And that can be unnatural and difficult, but when you can do that, I think it's a really rewarding life, because in my experience, acceptance and integration of both of these dimensions of our lives transcends either pure secularism or anti-intellectual fundamentalism. But again, this kind of work takes courage. And I'm so glad there's a society like this that'll meet up on Saturdays and talk about these kinds of things. And so again, just to acknowledge the Society of Catholic Scientists for these kinds of opportunities, UC Irvine for giving me the flexibility to explore these interests, and the parish that I go to in Southern California that has really inspired me. And that's it.